Hey there, everybody. Welcome to your MCAT channel session for this evening. My name is John Lennick. I'll be your host and tour guide. But as you know, we've got a couple other teachers in the room, which is why we have that Q&A as well as the public chat. So if you want to give a big what's up to Chris and Paul, they'll be helping you through all the little nitty-gritties that you guys want to talk about today. Speaking of, when is test day? I mean, this is full length number 5. You guys are getting kind of deep into it. We should probably have the talk. Desi and June, sweet Jennifer, so you're ahead of the game. It's fantastic. May 22nd. Nice Berenike. Perfect. So those of you that are testing, Sarah, I had a hunch that a couple folks would be testing in April because, of course, if you're 5 tests in, you're getting kind of deep into it. Make sure before your actual MCAT, you take all of that AAMC stuff. So, of course, now there's both the AMC sample tests as well as those question pools on your online syllabus. So definitely do all of those. If you're testing in May or in June, hold on to those for a little while longer. I'd wait about, maybe within the last month you can start doing those question pools and then really try to take that last AMC exam about a week before your actual real thing. Perfect. There we go. Yeah. So if you want to start with the question packs, especially Sarah, I'm glad that you're already getting on that right now. That's fantastic. So yeah, if you have any questions, let us know about that stuff. But oh man, coming in soon, and it's actually if you've already taken 5 full-length exams at this point, if you're testing a course in June or May, you guys are in really good shape. That's awesome. Ideally, we want to make sure you're taking about I'd say more than 8 full-length exams if possible. Sometimes that's not possible, but at least get I'd say more than 5 or 6, ideally. Cool. All right. So if you have any questions on that, hit us up. Remember, Chris and Paul are going to be running the Q&A on the right-hand side as well as they'll be talking a bunch in public chat. And I don't think I need to say this, but just as a heads up, if you have not taken full-length number 5, leave. I'm sorry for forcing you out, but you don't want to be here unless you've taken the exam. This is, of course, meant to be a review going through a lot of the questions that were in that full length. Now, as you know, this is not a comprehensive review. I know we've mostly been to these things before. We're just going to pick a couple questions and use it as an opportunity to talk about a couple different key concepts and a couple different learning goals. And so specifically, the learning goals for today are using information from the passage in, of course, the biochem section to effectively answer questions. So if you haven't done this full length in a while, a lot of this might not be so familiar because it's been a bit. So we're basically just going to pull chunks from the passages that you need to solve all these questions today. After that, we'll talk about, of course, making solid predictions, just to explain a given phenomenon. Some of these we're going to pull from passages too, but some of them will just, we can use outside knowledge to answer by themselves. And then my personal favorite aspect of this class is using data and figures to quickly answer questions. And so we'll go through a couple of things where we need the passage or where you just need the question stems image itself to go through and really answer a whole bunch of questions. Cool. All right, so that'll be the focus for today. Remember, it's not comprehensive, but if you have any questions at all over anything that was on your full length or test day based or basically anything you want to talk about, let us know. We are here to talk about. We're here to help any way that we possibly can. That's what MCAT channel is all for. All right, so we'll jump right in. We got a bunch of different questions to go through. We'll kind of just knock these off and give us an opportunity to talk about a couple different things. But this one came from pretty early in the test, question number 8 specifically. It says that your Duchenne muscular dystrophy does not affect smooth muscle because. So we need to actually figure out what the heck the specific type of muscular dystrophy is really referring to, and so we got to pull the information from the passage. Now to make this easier, and since you don't have the test in front of you, for all of this first learning goal, which is one of the shorter parts of today's class, we're just going to pull up the passage notations that we have right here. And so let me know, what's the key word in this passage that lets us know why it would not affect smooth muscle? Nice, Sarah. You got it. There we go, Jennifer. Same thing. The sarcomere, right? So tell me, what is a sarcomere? Well, let's do a little bit of content here. Oops, sorry for that. There's a couple ways you could describe this. Yeah, it's the individual contractile unit within a skeletal as well as cardiac muscle. You got it. So we call it striated. And exactly to tell that is totally true. So to draw out a very, very basic image of it, I'll draw like a couple different sarcomeres here. Let's do this real quick, and we'll give you a little bit of a quiz to kind of think about this stuff. But if I were to draw it as such, what would the black lines be? Actin or myosin? So we'll try to hit a couple things here today. I'll make that a little bigger. The black lines are actin. Why, Fong, why are you saying that they're actin? I did draw these intentionally to look like actin. Yeah, exactly. So remember, the actin is the thin filament. Exactly, you got it, Sarah. Mostly just because the actin will look thinner in the drawing, and the green or the black is going to have the actual little like heads that do the grabbing. The green lines are the black lines here. The way this is drawn. I want to use this as an opportunity to hit a couple content ideas. So what has the actual heads that grab onto the other strand and actually cause the contractile motion? That is the green, the myosin, Sarah, exactly true. So basically what that would mean is these little green bars have the little myosin heads that reach out and grab onto the actin there, and it's when those little heads basically go through their, what do we call it, power stroke, is when it's going to actually have contraction going on. And so the sarcomere, of course, is the functional unit of the muscle, and so, of course, here is one sarcomere right there. Of course, we could break it down to its component parts. Right in the center right here, you call this your, actually, do you guys know what that's called? We could do even these little breakdowns, since these are based in, of course, covering some cool concepts and ideas. Close, not M, very close to Tadel. It is another consonant. So between here and here, it's the H zone. Notice that myosin begins with the consonant M, and H is also a consonant. You can also look at the bands that are only actin. So that would be just from here to here. Do you guys know what the actin bands are called? I don't know why this gets tested, but it totally gets tested. You got it, I. And then the one other big one for you guys to know would be the one that shows the entirety of the myosin, including the overlapped. What's that one called? That's actually the end. So the Z disc or the Z line, that's actually the end of each sarcomer. So those are your Z lines right there. So this one's different. It's the entirety of myosin, including the actin overlap. It is the A band. It's the A band. Yeah, there you go, Sarah. There is an A. You got it. So here's your little mnemonic, just to kind of remember this stuff. H is myosin only. Both H and M are consonants. I is actin only, and of course actin A and I are both vowels. And then the A band shows all of the myosin, and it's also the only one that's always the same length. Notice I gave you a bunch of little A mnemonics there. So A is always the same, and it's all of your myosin. Both of those, of course, are A words. So little ways to remember. I don't know why this stuff still gets tested, but it totally gets tested still, and so it's one of those things that's just good to memorize. So of course, yeah, mnemonics are a nice way to remember those things that are, kind of weird that they test it a lot, but it's good to know. So. Cool. All right, so that's this one. So then, of course, what's the correct answer if we know that if we had sarcomere, we know sarcomere specifically for skeletal cardiac muscle because it's striated. Something about looks like answer choice. B. Yeah, you guys got it, Neva. Tadel, everyone's saying it. You got it. It's answer choice B. It's just because they're not organized into sarcomeres. They don't have that, of course. Also, I guess one more thing we can say. Smooth muscle, is it mononucleated or a polynucleated? Smooth muscle specifically. Smooth muscle is an important detail as well for test day. Yeah, it's mononucleated, Tadel, exactly true. So your smooth muscle is actually like mononucleated, whereas your skeletal muscle is actually, I don't know if they call it polynucleated or just multinucleated, but the idea is that it's a bunch of different cells that all fuse together to make one big functional unit. So all of the cells are basically fused, making it look like there's a whole bunch of different nuclei all together. Cool. All right, so a couple big details right there. All right, well, we won't go into content for all of these to that extent, but this was a really good opportunity to hit a couple of things since you are testing soon, that I had little mnemonics for you to help you guys memorize. So whenever there's a neat little mnemonic, I'll slow down like that and give you guys a couple details. All right, so next one, moving quite a long ways into the test. Now we're on to question number 21. It says this PRPCs, isolated from electrophoresis. We'll get into that a little bit later on today as well. But so when we move, we found that it mostly has this specific conformation. So this was the passage that talked about prions. And I don't know if you guys have actually heard about prion diseases before, but they're terrifying. It's pretty much the most terrifying disease ever because there is no cure, and if you get one, you're going to die, effectively. They're really, really scary things. So heads up about prions. All right, so basically we're looking for something, experimental modification, that'd be useful in isolating this native conformation. All right, so of course we'll have to pull some information from the passage. As always, we're going to make it easier by citing it for you. So what information did you need from the passage here? So notice we're comparing the native form and this that SC, the scabies or scabies. Yeah, there we go, Sarah, exactly true. So we know the primary structure is the exact same. It's the secondary structure that changes. They have that, of course, in the very bottom of the passage right here. It says the presence of it causes it to switch conformations, going from the alpha helices, which, of course, to the abnormal form, which is your beta-pleated sheets. We know these beta-pleated sheets are basically able to stack, which means they basically accumulate and call these aggregates that we like to call plaques. So notice here, we're talking about removing it, right? And so we talked about actually like using different types of solvents potentially to actually remove these. The biggest difference here, if we know that the beta conformation, actually, well, specifically, we went through this experiment as in the question stem. It says that the organic solvent, of course, is most likely is going to remove the PRPC, right? So now, if we want the native form, what would be, let's think about solvents. So we know that the beta-pleated sheets, the screwed up form actually is solvent, of course, aqueous, you could say, in organic solvents. So what would that tell you about this SC form, like in terms of its actual solubility? Polar, nonpolar? If we're using an organic solvent to extract it. Yeah, it's nonpolar, right? Which also makes sense because it says it's accumulating in plaques, right? So it sounds like a hydrophobic kind of thing. So we know, of course, this scabies form is going to get extracted by a nonpolar organic solvent. So then, of course, if we want to use something else to extract the other form, we would, of course, assume this one wouldn't be an organic solvent. It would be an aqueous solvent. And so the couple hints here is that we knew the beta form associated with organics. We're going to try to find something that's different here. Hopefully, aqueous at least would not extract out the beta sheets because we know that they're soluble in that organic solvent. Additionally, whenever it says something like that, as it says in the passage, in terms of them basically stacking, right, so they're going to stick together, that's a good hint for hydrophobic, as well as accumulating together, giving those plaques. So, of course, it's the same when you think of like heart disease, right? If you eat too much cholesterol, all that cholesterol, also very hydrophobic, is going to basically conglomerate together, making plaques, and then get stuck in your arteries, causing heart disease. And so it's the exact same idea here. We tend to get plaques from things that are very nonpolar, like to stick together in the aqueous environment of our body. All right, let us know if you have any questions on that. Polar versus nonpolar, aqueous versus organic. Crazy high yield for test day. If you have any questions, please just hit us up in the Q&A for that one. All right, we're going to go through a couple of these different ones. This is from the same passage, as you can see. So let's go a little further. It says, how's translation expected to differ between these two different forms? So once again, let's do the foundation. Translation means what? What is, of course, translation? Turning one to one. Perfect, Sarah, you got it. So RNA into protein. Perfect, yeah, specifically mRNA, right? So it's your post-processed mRNA, right? It's the actual full thing. And so what do we know that was the same about the scabies form versus the other form? Or scrapey, sorry. Scrapey form. Yeah, Jennifer, exactly. It said the primary structure is the same. So if the primary structure is the same, how's translation going to be different? It's not exactly. It's going to translate it the same way. What would be different? What would be different? Yeah, one thing is it's just not going to be the same. Yeah, the post-translational modifications. And if there's one protein, I would say, that you want to be aware of that basically helps control the post-translational modifications, which is primarily the folding, but also you can glycosylate and do lots of other things. What's that protein that's associated with letting proteins actually fold up properly? There's a certain component that actually lets things fold the way they're supposed to. This is one you do need to know the name of. Nice, a chaperone, Sarah. Perfect. So chaperones basically allow these proteins to fold effectively. If you're missing a chaperone, it might fold incorrectly, which of course then could cause major problems. Potentially nothing as bad as a prion, but in this case, this is basically what happens. And so if there's one thing to take out of this passage, it's to never eat brain tissue. The vast majority of the ways that we actually get prion diseases is from eating the nervous tissue of various animals. And so don't eat brains. That's your take home for that question. Because there is no cure for prion diseases, so we better just, of course, avoid that altogether. All right, so we're kind of jumping around, pulling things from the passage. Here it says, moving quite along, so this is question 42. Information in the passage tells the retinal pigment epithelium should have above average numbers of. So we got to know basically more about this retinal pigment epithelium to know about their lysosomes, mitochondria, or ribosomes specifically. All right, so there's a little abbreviation. That's your retinal pigment epithelium. So one of these should stand out pretty quickly, that either be correct or not correct. And let me know what you're noticing in the passage there to let you at least choose one. There's two keyword phrases that I noticed. I'll see if you guys see the same ones. Yeah, Mareniki, that was the first I noticed when I was looking through this as well. We've got phagocytosis going on, right? So it means, yeah, we're recycling, which organelle, of course, is highly associated with, of course, the breaking down and recycling. That's your lysosomes, you got it. They're like the little recycling plants that exist within your cells. You got tons of lysosomes that are, of course, going to break all the stuff down and then recycle it for use. You got it. And so since we know lysosomes definitely needs to be true, you can get rid of C. That doesn't help too much. And Tadel is hitting the other key point. So notice not only we do know that lysosomes are going to be key, it also says in the passage that they provide glucose and certain lipids to rods and cones. And so if we've got a type of cell that's providing glucose to other cells, what do you think about these answer choices? Yeah, what about mitochondria, Frankie? Tell me more about it. Good thing, bad thing. Or mito-C, as they say in that thrift shop parody song on YouTube. So, yeah, Tadel, exactly. So the whole idea is if it's going to provide glucose to other cells, if it had above average amounts of mitochondria, that would mean that it would use up glucose like crazy. It's the same reason that you don't have any mitochondria in what other type of cell? To not use up the good stuff. Red blood cells, exactly. It's not for glucose. In this case, it's for oxygen, but it's the exact same idea. Red blood cells' job is to, of course, transport oxygen. If you've got mitochondria around, it's going to burn up O2 and it's going to burn up glucose. And so in this case, we know that we don't want to use up our glucose, so you don't want to have mitochondria. And if you can knock out 2, saying there's no mitochondria, notice that it gets rid of D. It also gets rid of A. You're left with only one answer. It's got to be answer choice B, without even having to look at answer choice 3 there. So that's the fun way to do this. You don't have to go through everything. All you have to do is do enough to actually eliminate the answers that are incorrect. Cool. Nice. Oh, gotcha. There you go. Cool. That totally works. Yeah, you got it. As long, right answers are right answers. That's really all that matters. It doesn't really matter how you get to it. A point is a point on test day, as we like to say. Cool. All right, we'll go to the next one. And let us know if you have any questions on any of those. All right, so now we got into myasthenia gravis, experiencing rapid fatigue before muscles are able. So, okay, so muscles are unable to sustain contractions, right? And then we want a plausible explanation for this. So now, one thing I do want to say, you got to know a couple of things about muscles. But so, what's associated with muscles contracting? There's a couple of things that you want to know. Just you guys tell me the background, and we can kind of play with this a little bit. And yeah, so tell me about the calcium. What's calcium do? Calcium is the controller. I totally agree. It's what basically stimulates the contraction. There's a couple things we can say here. You can say where it's stored, what it binds to. I don't know if you guys know. So there you go. You got it. It binds to troponin, which then moves tropomyosin out of the way, so you can actually have your power stroke. Cool. Calcium's also associated with the action potential, Tadel. Remember, calcium's necessary for the vesicles to fuse with the presynaptic membrane. That's also totally true. Oh, cool. Thanks, Peter. Actually, do you mind swapping that out? One of the other teachers. That way, it'll make it easier. It's fun. Oh, thanks, Peter. Cool. So that's one thing. Calcium is necessary for the muscular contraction. Second thing is, what is the neurotransmitter that goes across the neuromuscular junction, the NMJ, as we like to call it in the biz? Nice, Tadel. Acetylcholine. You got to know that one as well. That's very, very important. And so basically, if we are not able to sustain contractions, I would assume, this is making our predictions one of two things, that it's probably going to relate to either calcium not being around, or of course that there's some kind of problem with acetylcholine. That would be your first two kind of anticipations as we go through it. Cool. And so without even using the passage, we could probably answer this with outside knowledge. Which one looks closest? I see A talks about calcium, B talks about acetylcholine. Yeah, I agree. What's the problem with A? Even though, of course, it talked about calcium, it would be an... Yeah, exactly. It says increased calcium, right? A decreased calcium, yeah, that's possible. Increased calcium, that's going to be an opposite. You got it. So no good there. Cool. Nice work. Perfect. So this is the kind of way we want to do most of these questions on the test is when you get these little cues in the question stem, it's like, all right, we're talking about muscular contractions. That means we're talking about, I guess, of course, the output, right, would be your efferent neurons you can get into. You can get into acetylcholine being the neurotransmitter. You can think about a calcium. You want to start priming the pump, so to speak, of all those different foundational content that we really think about when it comes to muscular contractions. All right, so this one's weird. The odorant receptors mentioned in the passage are likely, so this we definitely need to pull information from the passage. This is from near the end of the test down to question 54. And so this is some important background knowledge as well. Tell me what kind of stood out in the actual question stem here in terms of helping us figure out what kind of receptors these are. Yeah, CAMP's the big one, Mareniki. You got it. There we go. So we've got CAMP, we've got a signaling cascade, and we got GTP dependent. Those three things together should trigger us to think of a very important type of membrane receptor. Do you know what kind of receptor would be associated with those three things? Nice, Jennifer. There we go, Sarah. Let's just say the same thing, the G protein coupled receptors. Problem is, none of the answer choices directly say G protein receptors. So you got to know a second characteristic of your G protein receptors, and what answer choice gives you the other characteristic to know? They are snakes, Sarah, exactly true. There are 7 transmembrane components, exactly true. Answer choice D. So notice, not only did we have to use our outside knowledge in pulling out from the passage that it's G protein, the actual answer choice, you got to use your own outside knowledge to know, okay, well, these G protein coupled receptors have, of course, these the 7 transmembrane receptors. I like that Sarah said they're like snakes. Totally true. They loop in and out. Totally true. Good. Very good stuff here. On a roll, as we said. All right, so that knocks out the very first learning goal. This second learning goal here, we'll move through this one a little bit more quickly and kind of talk about a couple given phenomenon and things to know. We're not going to have to pull passage citations as much for this, so it'll move a little bit more quickly. All right, moving to the next one. Let's talk about fertilization in humans. It says the entire sperm's absorbed into the ovum, but the sperm mtDNA, what the heck is that? mtDNA? Mitochondrial DNA, exactly true. So now, wait a second. How is mitochondrial DNA different than the rest? Why do we have to give it its own designation? What's the deal with that? This is actually super high yield. So it's circular, it's self-replicating, exactly. It only comes from mother because mitochondria is kind of like an ancient prokaryote that our cells absorbed a very long time ago. So it actually has its own DNA. Now, in reality, since we've had mitochondria for the past billion-ish years or so, a lot of the genes that mitochondria needs has actually been incorporated by our own genomes, but it still does have its own DNA, and exactly, it looks like bacterial DNA. Totally true. Also, we only get it from the mom. Good call there. So now, but since we're absorbing the entire sperm, does that mean sperm doesn't have mitochondria? No, they have lots, because of course they swim. That's exactly true, Peter. They got to swim all the way through the entire body, and think about the distance they're traveling for being a single cell. Holy crap, they're going really far. And so they got a lot of mitochondria, but for some reason it doesn't show up in the baby. So what would you predict? As we know, doesn't leave it in there. Stuck in there. DNA can't get out. So what would probably happen to it? Bingo, Tadell. We got to break it down, right? So since, of course, we know that it's not there, which means it must be destroyed, we got to find some kind of answer choice that relates to it basically being destroyed. Any answer choices seem to be close to that? Oh, C, yeah, you got it. C actually directly says that. Targeted and destroyed by maternal organelles. Nice work. Answer choice C. Good job. All right, a couple other ones here. This one we might need to use our own knowledge, unless if you know about shingles. So remember, what's like the virus that precedes shingles, which a lot of us will likely have to worry about, unless if you were born after the point that they started vaccinating. Chickenpox, exactly. At least, I don't know if you, when I was a kid, they just made us get chickenpox. There wasn't a vaccine yet. Did you guys get a vaccine, or did you have to get chickenpox when you were tiny? Just out of curiosity. I know it was actually not too long after I was Yeah, exactly, Jennifer. Same thing. So that's the problem these days is that most of us we were just told to get chickenpox, so you would then be immune to it. And then of course the problem is now we are now susceptible to getting shingles later in life. So now there's a couple things to know, just especially going into medicine about shingles. But so the skin pattern, I'll give you guys the passage. But I actually would recommend you guys. Yeah, totally. I know a lot of people in their 50s and 60s have had it recently. But there's an interesting pattern about shingles. Namely, it's on the single side of the body and tends to form in a single strip, either in the face or on the body. You often get it down the back and things like that. But the key is it's in the single side. That's very representative of shingles. So why is that? There's also a key word for you to know about this too. It's localized, so an answer choice has to deal with something with being localized. That's the key. Actually, let's look at this. So C says localized, and another one talks about being localized as well. There's two of these that seem to do that. There's one other answer that does talk about being localized. A, Peter, exactly true. So either C or A, these are the two that do relate to it being basically localized in a single location. Now, the thing to be aware of, and you can pull this from the passage or, of course, use your own information, but notice that it becomes dormant in the cell bodies of neurons, specifically. It's where this thing lives. I believe herpes simplex, like the oral herpes one, is the same thing. It actually lives in the neurons of whatever's close to people's mouths, which is why it actually blows up in the same place, because, of course, it's actually nerve-based. And that's also, as Aislyn's saying, that's why it's so painful. Oh, my gosh, someone with shingles, like, it hurts to touch, it hurts to move. It's really, really painful because it actually is a nerve issue, which is why answer choice A is actually going to be better than answer choice C because it specifically said it stays in neurons, and so it actually affects the skin innervated by that one nerve. There's actually a special word for this, the skin that's innervated by that single nerve. Do you guys know what that's called? This is a good term for you to know as well. I'll put it in the chat if you guys don't know it. I think someone's going to get it, though. I think Peter knows it. It is it's okay. It's a dermatome. You guys heard that term before? Notice derm, skin, right? So it kind of relates to the same idea. Probably heard it before. Maybe not. That's okay if you haven't. I guess, yeah, okay, cool. I figured if in some physiology classes they'll talk about it and some they don't. But that basically refers to that specific region that's innervated by that one nerve. So it's the dermatome. Cool. Background knowledge for you. If you want to geek out on more physiology about that as well, hit us up in the Q&A. We could talk for days about this kind of stuff. Oh, gotcha, homunculus. Yeah, remember, that's the region in your brain that kind of represents the actual, well, you can actually have sensory or motor homunculi in your brain, technically, but it's the weird-looking human that shows where all the nerves actually go to, which is why the homunculus has massive lips and face and hands, and the rest of it's pretty small because you got tons of nerve endings in your hands and your face and your lips especially. That's why kissing's so much fun, right? It's all about the homunculus. All right, so next one is question number 31. It says, which of these explanations for the white colony on plate 6 is most likely? So this one, we're definitely going to need some information from the passage. We got to remember what the heck is going on with the white colony, plate 1, plate 6 specifically. All right, so this was one of the more intense passages. It's kind of cool. So we basically got this talking about, this is the beta, what do you call it, the lac operon passage as well as ampicillin resistance. But so we got this thing that says we've got this plasmid where it's basically, we have I won't get to the plasmid yet. Basically a minimal medium with lactose and ampicillin, and this X-gal, which basically turns blue when it's been cleaved by beta-galactosidase. And then after 24 hours, we basically look at this chart here. All right, so specifically looking at plate 6, as you can see, we get primarily blue colonies. And so when it says blue colonies, what do we know about this cell? Because we gave it the X-gal. Remember, it said specifically that X-gal turns blue when it's been cleaved by beta-galactosidase. Exactly, Sarah, you got it. Which means they've actually got beta-galactosidase, which of course is associated with LAC-Z. Totally true. So that means that the blue colonies have beta-galactosidase. That white colony does not. So what answer choice looks closest to that? You got it. They can't make their beta-galactosidase, so of course we're missing LAC-Z. It must have lost function in, of course, the beta-galactosidase gene, which is your LAC-Z. Nice work. These types of passages are so freakishly popular where they talk about plasmids, conferring different types of resistance, and then of course you need to interpret what kind of bacteria it's gotten given basically what kind of growth happens as well. I'm sure you've seen already a bunch of these, but super prevalent. If you guys want to talk more about ampicillin, LAC-Z, beta-galactosidase, plasmids in general, hit us up in the Q&A. We can get really into that stuff, so just let us know. Cool. We will come back to this passage a little bit again later on, too, I believe. We have one more question on it at the very end. All right. So moving back a bit. Oh, actually, no, this is the same one. Nope. This is a different passage, actually. So this one, we got experiment 2. Once again, pull it from the passage. It says that this guy in the 1800s basically had this cell embryo, there we go, shook it really well, and then basically every cell developed into a complete, though smaller than normal embryo. Okay, that's kind of weird. So first of all, what do we expect then? So basically we still got the same number of cells, nothing died, but everything's going to be smaller. And so if we've got the same total number of cells, they've been split apart, but they're smaller, what's the smaller actually hinting? And that's kind of the key word in this question stem. If everything's going to be smaller than normal, it's the only thing that's different. Yeah, the growth phases. Nice work, Sarah and Tadal. So of course, when we talk about the cell size, remember it's only in G1 and G2 that the cell actually gets bigger and bigger and bigger. And so the actual rate of cell division is going to be the same because we have the same number of cells. Everything's pretty normal. It's just that the stuff is smaller. And so it definitely will, of course, be G1 and G2 that are primarily affected there. Nice work. So notice this is mostly an opportunity to kind of hit a couple key concepts and talk about it. And so that's why we like these test reviews, because it's not just going through your test, it's kind of point out some key details there. Yeah, we don't need to go. I think you guys are, if you have any questions on synthesis, mitosis, hit us up in the Q&A. I'm assuming since we're all at test 5, we're pretty good on the basics of cell cycle, but don't hesitate if you do have any questions at all. All right, this is another kind of weird one, same passage. Best explanation for how these organizer cells can still function even after they've been killed. So this was in the passage. It told us specifically that these organizers, basically, even though they're not alive, they're still going to induce development of different types of structures. So a dead cell is somehow inducing development, which is interesting. So then what do you know about the metabolic activity of these dead organizers? So if they're dead, what are they definitely not doing, first and foremost? A couple things, actually. I'd say several things. Yeah, they're not growing. They're also not transcribing anything, right? They're also not producing mRNA exactly. Yeah, exactly. So they're not going to, definitely true, Sarah. Not good answer choice D. There's no metabolism going on. They're dead. They're no longer functional. But they're still inducing development. So what do you think, how could they potentially be inducing development if they're dead? They're not making anything new. They're just being there, and what are they probably doing? Because in order for them to induce development, they have to be acting on the other cells nearby. So how would they do that? There's a keyword here that you want to know about inducing development too. Do you guys know what that keyword is? Basically how one cell can direct the development of another cell. Yeah, you're getting the right idea. Tadell's saying it, totally true. They're adjacent. I was getting at transcription factors. That's a familiar term for all of us, correct? Basically, it could still be releasing components. It's not making anything, but as it leaks out, different transcription factors could still be going into its environment. And so adjacent cells, as Neva says, they're totally true as well, can still induce development and whatever it might be. So basically, as this thing is decaying, it's still releasing some kind of compounds, which of course are then causing some kind of change. And what answer choice looks closest to that, at least? You got it, answer choice A. So basically, it's acting as some kind of container, and so it's, of course, as it's breaking down, it's still releasing different signaling molecules, primarily transcription factors is what we call them, but the very generic word would just be, signaling molecules. Cool, which then, of course, cause transcription in different types of cells. So what's the problem with C? Great question, Renke. What's a problem with C? You guys can answer that one, maybe. Yeah, you got it, Jennifer. It's not that it's inhibiting. It is actually causing differentiation, right? So it's not inhibiting signaling. It's actually activating signaling between different molecules. That's kind of the key there. It's causing development, you could say. Cool. Yeah, it's a trap opposite. And that's the thing with those opposite answer choices. They generally have the exact right language. They just switch one thing around, and then it makes it all different, you know? Exactly, yeah, totally. Yeah, as Paul says, exactly. That's the problem with those opposites. It's one word that we read, a little quickly, and it makes it all wrong, even though the language is pretty similar. All right, well, we'll go through this one pretty quick, and we'll jump into I believe, our last learning goal. Yep. So which of the following is least likely to be found inside a viral particle? This is a good one. What are viruses? Let's kind of get into viruses a little bit. Yeah, agreed, Sarah. Super short questions, super short answer choices. These are my favorite, especially in the bio section. Oh, man. So they are, well, yeah, you guys are right. It is C. But tell me more about viruses in general. You're right. It is C. Viruses don't really have lipids. But what do they have? So they're protein coats with nucleic acids inside. What kinds of nucleic acids? You got it. They can either be DNA or they could be RNA. They could be single-stranded. They could be multiple double-stranded. They could be multiple strands. In fact, I don't know if have you guys heard about the Pandora virus? A fairly recent discovery. I believe we found it frozen in Arctic sea ice. Freakishly enough. Yeah, totally. Sense antisense too. Totally true. Oh my gosh. So basically they discovered a virus. It's a whole category of viruses. The Pandora virus was one of the first ones they found, so they named it because they're like, oh my gosh, this could be a box of worms. That's bigger than small bacteria. A virus that's bigger than small bacteria. The thing's got hundreds of genes, and it's a virus. And apparently, they're all over the place. It's not just frozen in Arctic sea ice. You're basically breathing them right now, apparently. But the problem is, yeah, I was going to say apparently they're super common, but what it seems to be is that they only affect amoebas that we know. So they basically breed using amoebas. They don't seem to affect eukaryotes so much, so we don't think they're a big concern. And that's the weird thing, Sarah. They recently just found it. I think the problem is that we didn't look for it because we didn't think there could be a virus that was that big. So like anything that size, we looked for, like basically not symptoms, but signs of, prokaryotes. So of course division, all that, and it wasn't. But yeah, it's pretty interesting. If you just do some Google searching of I forgot the name of the genus, but it's basically giant virus. I guess it's not a genus because they're not alive. That's the other thing to remember as well. Remember, viruses are not alive because they're not self-replicating. So that's a very important part. But I agree, Chris, it does sound like something from Alien. Maybe they're just all lying dormant, waiting to get a signal from space, and then they'll basically take over us all and we'll turn into aliens. Who knows? But it doesn't really matter either way because it's too late now. They're already here. All right, so you guys got it. There's giant viruses now, be aware of that. And so we call that protein, there we go, that is the category. And so, of course, the protein coat we call a capsid. And really, viruses are very much like spaceships. They land in a cell, they inject their genetic material inside the cell, and then the cell basically picks it up and then does things with it that it needs. Yeah, yeah, that'd be pretty sweet. That would make sense to tell. Not sweet, but you got it. Maybe they gave us the viruses. Who knows? Who knows these days? Movies give us every possible idea, so I'm assuming at least one movie's got to be correct. Maybe. All right, so that brings us to our third and final learning goal for today. This might be my favorite learning goal, where we get to utilize different types of data, figures, charts, graphs, all that kind of fun stuff to answer a couple different questions. And so we only have a couple of questions here, and so we can kind of take some time with it and really kind of break them down. And there's one real tricky, weird one that I want to spend extra time with. But first, let's do this. First question, the entire test. In order to answer it, you need that giant image that was from your test, which shows a little tree of life for different organisms. Specifically, it looks like mammals. All right, so now it says, which of the following is least likely to share a common ancestor with bats and whales? So first thing we got to find, of course, is bats and whales. Notice I've already got it highlighted right there. So notice, I guess, what are you looking for when you want to find something that's least likely to share a common ancestor? Yeah, exactly. Something that's further away in the tree, not necessarily distance, but the most branched away from it, exactly, you could say. Totally true. So now notice here that we've got, basically the way that you could look at this is we'll look at bat. And so if you look right here, see, this would be the common ancestor that eventually led to a bat. So not only did this common ancestor lead to bats, it also led to everything that's down this chain, and this chain here. Does that kind of make sense? So like you find the actual juncture in there, and that's where like the common ancestor is that we don't have a name for. It went extinct a long time ago, but it eventually led to that. Cool. There we go. All right, so we know that it's at least not going to be anything that's linked to that central ancestor. So horse, definitely not. Rhinoceros, definitely not. Cat, dog, bear, weasel, definitely not. So it looks like we can get rid of horse, we could said. We can get rid of cat as well. And where's seal at? Yeah, right next to cat. Well, if seal's right next to, there it is, right there. That means, of course, we can eliminate that one as well. And so the correct answer has to be A. It's got to be a shrew. So a shrew is much more like a vole or a mole. It's a little pointy-nosed little thing. Yeah, I agreed. Voles are actually adorable little creatures. You tend to find them quite a bit. But so that is the whole idea of different common ancestors there. Find the juncture in the tree. There must have been something right there, and that led to all of them. I actually saw a pretty hilarious infographic recently that talked about misconceptions in evolution, and basically what it like the image had, it was like evolution doesn't work like this, where it was like fish, gorilla, and then human. Just as it doesn't work like this, where it's like you have like grandpa, which leads to mom, which leads to you, and that's kind of like one of the big misconceptions. Did anyone see that infographic? It kind of came out recently, but it was talking about people who basically are using evolution incorrectly. It's that like it's not like your grandma then reproduced to make mom and then that made to you. We have a big family tree that basically branches all the way down. It's the same thing with evolution. It's not like basically monkeys turned into humans. It's that there's a common ancestor somewhere in the back burner way down the ways. If you want to look it up, I'm sure you can find it online. I saw it recently. But it was a very witty infographic about evolution. And I figure since we're all scientists, we would probably enjoy things like that. And yeah, Jennifer, that's the problem with a lot of these graph interpretations. And so that actually brings up a really good point when we have these. There is so much information in the chart that you don't want to look through the whole thing. Instead, you want to identify different answers and flag them. Be like, okay, there's shrew, there's cat, there's seal, and then eliminate things that are there. And yeah, seal was on the same thing as cat. I couldn't see it either, as you guys noticed when I was going through it. Find out where it is at the very end there. So gotta find it, gotta eliminate. And if you're not sure, remember, if you're short on time, narrow it down to seal and shrew and pick one of them if you need to. Try to avoid guessing randomly between the last two unless you have to, but sometimes that's really just the situation that we're stuck in. All right, so we'll do the next one. This is another very common infographic, I guess you could say, that we see on test day, these pedigrees. And so if you ever see a pedigree like this, there's one thing that I want you to notice first. There's two things, but one thing I want you to notice first. I agree about the squares. First, I would say is that it skips generations. This would actually be the most important thing to identify. If you ever have a trait where you have a generation being skipped. Now, it doesn't need to skip an entire generation there. Notice that the exact same thing happens over here because you have two parents that are unaffected and then children that are affected. Now, it doesn't matter if it actually skips the entire second generation or not. If you ever have two parents that are not affected and children that are, what does that mean must be true? It's got to be recessive. You got number one, it must be recessive no matter what. I don't care what kind of recessive, it's going to be recessive, which means you can get rid of B, not dominant, get rid of D, not dominant. Now, also, Sarah and Jennifer and Mareniki are pointing out another very important detail. It seems to me that only males are affected. And what does it mean when only males are affected? You got it. It is X-linked. It is sex-linked. So remember, sex-linked traits are X-linked traits. Why is it that males get affected so much more than females when it comes to these sex-linked traits? Exactly, because we only have one X, females have two. And so the whole idea is that a female can be a carrier to sex-linked traits because she can have a single X that affected and another one that's not. Males, on the other hand, there's no such thing as a male carrier for sex-linked traits. They either have it or they don't have it. Females can either not have it at all, they can be a carrier. Is it possible for females to be affected by sex-linked traits? It is. In order for a female to be affected, she has to have double copies of it, though. That means that she had to get it from her mom, and she also had to get it from her dad. So real quick to talk about this, if you are a male, you have to worry more about sex-linked traits like male pattern balding. And so if you are a male and you're wondering if you might go bald, who should you look at? Who is the person you need to be nervous about being bald? Who's great? Which side? Totally true, Jennifer. Which side, though? Your mom's side or your dad's side? There you go, Aislinn. You got it. It's got to be your mom's side. Because if you think about it, if you're male, you had to get the Y from your dad. If you are male, it does not matter if your dad is bald because you don't get an X trait from him. So all you have to do is look at your mom. Well, if your mom's bald, then sorry, you're screwed. You're definitely going to be bald too. If your mom's dad is bald, what's the chance of you yourself being bald if your mom's dad was bald? 50%, nice work, because the idea is, if your mom's dad was bald, well, we know for sure that he was affected by the trait. And if he's affected by the trait, since we know you're getting the X trait from your dad, that means, of course, that you must at least be a carrier. And if you're at least a carrier, that means, of course, that there's a 50% chance you get the carrier gene, there's a 50% chance you get the other gene. So you got it. It is a 50-50 shot. Unless, of course, if your dad was bald and your mom was a carrier, then there's actually a chance that the female children can also be bald. That's why, of course, if you're nervous about your kids and you're a female, well, if your dad is bald and you happen to marry a bald man, then there is a 50% chance that your daughters will actually get male pattern balding. Mostly it shows up as just being very thin hair from what I hear. So remember, selective breeding habits are very important. You don't want to mate with just anybody. Make sure you check out their genome first. Very important. All right. That's why it is, of course. There we go. Right. See, real life tips in MCAT world. All right. So, of course, X-linked recessive. I apologize if you're dating a bald man and your father was also bald. Don't worry about it. You'll probably be fine. All right. Let's do the next one here. I was going to say, I try to give you fun facts because, of course, yeah, exactly. It's really important to think about this stuff and how it relates to the real world. That's probably the most important thing here. All right, so now if there's one thing to take out of this question, it's about recombination. And what do we know about recombination in genes in general? Probably the biggest point. It's about a distance thing. How do they relate to distance? Ooh, actually, Jennifer, other way around. So it's actually the further they are, they're the ones that are more likely to recombine. All right, so now here's the idea here is if we notice these different traits right here, for example, B and D are 20%, that means, sorry, A and B are 20%, that means A and B are actually pretty close together. Whereas if you look at A and C, we know A and C are a little bit further apart. This is a tricky question, so I wanted to spend a little bit of time with this one. All right, so now what we basically want to do is try to map this out. And so we know that A and B are going to be kind of close together. We also know that C and D are going to be kind of close together. We just don't know really the order between them. So now notice if you look at A and D is 30%, B and D is 50%, what does that mean about the relative locations of A and D? Closer or further? A and D must be a little bit closer together. Exactly. I think I actually randomly drew this out exactly how it's supposed to. A, B, and actually, there we go. You can tell from what we just said there. We know C has to be a little bit further away. Because notice we know A and C are actually pretty far apart because we have a higher combination frequency. So between C and A versus C and D, we know C does have to be further. So of course it's going to be further out there to the side. I happened to draw it almost exactly. I switched A and B, but pretty darn close. So now here's the thing. We want to now figure out what the recombination frequency for B and C is. And notice B and C are the furthest apart from each other. So how do we actually calculate recombination frequencies? Nice to know, we add them up. So there's a couple ways you can do this. This is the way that it has right there. You could just say A to B plus A to C. But really what this means is just calculate the distance between. So between C and D, we know that there is a 20% chance. Between D and A, we know that there is a 30% chance. And between A and B, we also know that there is a 20% chance.
Cool. Good question, Sarah. I'll let Chris grab that one. In terms of actual practice, I wouldn't be surprised. We tend to use a lot of kind of like shortcuts and, you know what I mean, to solve a lot of this stuff. We'll see what Chris has on that one. It should be, yeah, so that's a good point. I was like, yeah, actually, that's a good point. So I guess the answer here is 70% to use the actual numbers right there, but I totally agree. I was just jumping into the math, and the actual exam wants it to give you 70 as well. But in actual practice, I agree. You're never going to get really more than 50% in terms of the actual application in terms of statistics. So we're going to skip that one for now. So mathematically, this is what we want you to know. You basically add them up as Tadell says. In reality, things are more complicated than that. And so we won't get into the actual reality because especially when it comes to genetics, things are always going to be a lot more complicated than it seems as a general rule. So yeah, that's a good point though. I didn't even think about that until we just talked about it. I was like, oh yeah, that's a good point. Really, Jennifer, interesting. That's so what probably happened was they changed it because that makes sense. It was probably recently edited because you can't ever have more than 50, and so if you're more than 50, it's going to be 50%. So agreed. It's like you would do the math for 70, be like, oh yeah, 70, but in reality it never breaks 50, so you just choose 50. So yeah, I'm glad it was already edited on your exam. So this mistake has already been caught. We will very soon update it on this slide as well as the exams that have not yet been edited. So now it's good to know that some are 70, some are 50. They're probably in the process of actually swapping it out. But no matter what, you kind of get the same idea. You know what I mean? Between the two. So let us know if you have a question on that. Hit us up in the Q&A. We could talk about that more. But before we do that, there is one thing I want you guys to notice about this question right here. This is the same chart that we gave before. And so notice here, yeah, and as Chris says, totally, the concept's the same. When we look at this table here, we want to find, of course, the percentage of stock plasmids that became hybridized through trials 1 to 3 is closest to. So we have to look at the trial, of course, that has both the stock and the hybridized plasmids to figure out, of course, which ones are going to be basically taken up the most by these different types of bacteria. And so which trial specifically do we want to look at here? What trial will actually let us know? Yeah, you got it. It's got to be trial 3 because in order to tell whether it's picked up that X-gal, or sorry, the actual LAC-Z gene is if it actually got that X-gal. Because remember, if it did have X-gal, then of course it's gonna turn blue, which is how we actually know. And that's what the difference is, whether it gets blue or versus white. So now what this means now is if it's the blue colony, then we know for sure that it got the LAC-Z gene, right, that we needed. Whereas, of course, if it's a white colony, it means that it didn't get it because it's not turning blue. It's not chopping up it doesn't have beta-galactosidase, I guess we could say. So what that means then is, this is the tricky part. You got to actually pull this from the actual data in the test. But remember what it said specifically was that we actually used the restriction enzyme that cleaved in the middle of that LAC-Z gene. So if we did get the hybridized plasmid, it should have spliced in the middle of the gene, which means that gene's going to be non-functional. And so if that gene's non-functional, it should be white, not blue. So this was passage-based. This one was pulled directly from there. If you want to check it out again, feel free. But the whole idea is if you have a restriction enzyme cleave in the middle of your gene, it will not produce that gene because it's been chopped in half. That's really the problem right there. And so if it did get that, of course, that means that it will be, in fact, white, which is then to get a percent, all you do is you take your 9 divided by the total number there, which, of course, brings us to 23%. So that'd be 9 divided by 39, which gives us that. Actually, there's only B's pretty close to that. And when you get math like this on the test, you don't have to actually calculate what 9 divided by 39 is. Really, all you have to do is say, okay, it's going to be a number that's kind of big, maybe like 20-ish or something like that, like round it, you know, from 9 to 10, 39 to 40, somewhere around 25-ish%, and B is pretty darn close. All right. This question's kind of wonky. We talked about this actually a lot before class because this, as you might have noticed, is not standard electrophoresis. So just as a heads up, because in electrophoresis, we don't have A, T, G, and C, right? You just have different wells. You like put a bunch of DNA in this well, you put a bunch of DNA in this well, you put a bunch in this. And what is electrophoresis basically? I was going to say, so it's kind of a little bit of a typo here, but we'll make it work. What does electrophoresis separate according to? What does electrophoresis separate according to? On size, exactly. So what I want to do here, I'm not going to get too much into the question itself. I want to hit the key concepts that the question is actually getting at. So now, if we know that we're separating according to size, what part of this is going to have the biggest chunks? The top part or the bottom part? The top part, exactly true, because notice if you, well, so here's the idea. Note DNA migrates down. Remember, DNA has a negative charge on it because of its sugar phosphate backbone. And so the further something travels, thus the closer it moves towards the anode, and yes, it's a positive anode because it's an electrolytic cell, that means, of course, the smaller it is. So we're going to have the big stuff up top. We're going to have the small stuff on the very bottom right there. And so now the other idea that we want to, of course, bring in here is that we're going to add a dideoxynucleotide triphosphate. So if you're going to add a dideoxy, so there is no hydroxide in a 3' location, what's the key concept they want you to realize here? If you had a deoxy in the 3' position, what's going to happen? It's going to end. It's going to terminate, right? Remember, DNA always goes from the 5' to 3' direction because you need the hydroxy on that 3' end in order to actually form the next bond. And so that means if you add a dideoxynucleotide, it's going to cut off the chain. So that means the very smallest chunk is going to be a T. So then where would you expect T to be? If we know that it is this very smallest chunk, would you expect it to be on the 5' end or the 3' end? Bingo, Tadell, it's got to be on the 5' end. Because that means the very beginning you have a T, and turns out the only thing you added was dideoxy. So you just get a nucleic acid chunk. So right off the bat, you know that it's going to start with a T, which eliminates answer choice D as well as answer choice A, because, well, D you can get rid of because it has U's in it. We're looking at DNA there, so that's the other way to knock out that one. Then, what's the next thing going to be? So the first base is going to be T. The next base is going to be what? Another T, exactly true, Tadell. So that means, of course, it's going to be T and then a T. The only possible answer choice is C. All right. Fantastic work today. That was the key concepts that we want you guys to remember in electrophoresis. Remember, the bigger the chunk is, the slower it moves. And, of course, if you ever have a deoxy in that 3' end, of course, it means you have going to terminate your nucleic acid. All right, so stick around. Kristen is up next for further a full-length 5 review. I will see you all in future channel sessions. Have a great rest of your evening.